Saber Masmoudi is professor of human genomics and head of molecular and cellular screening process laboratory. In Tunisia, he actively participates to the implementation of several technologies for human genetic molecular analysis, such as microsatellite whole genome screening, microarray, and next generation sequencing. And in this context, he will share with us the Tunisian experience of his laboratory and the application of next generation sequencing in the study of hearing loss, advantage, challenges, and the recent extension of this activity to neurological disorder. So you have the floor, Professor Saber Masmoudi.
Thank you, Shanez, for this presentation. So, do you see my slide?
Yes, but you can have the full. Yes, it's okay. Great.
It's okay. Thank you. So, thank you for this introduction and presentation. Also for having associated us as genetists with your collaboration with Professor Holden. So thank you, sir, very much, Professor Tricky, for this. You presented the NeuroGen, so this is really an association between neurologists and genetists. I would also thank Professor Holden and Dominic for the excellent presentation, and all participants for their presence. So I just come back to the first slide. So my presentation entitled Next Generation Sequencing and Facial Recognition to Improve Diagnostic of Genetic Disease in Tunisia. So as you mentioned, currently my institution is the Center of Biotechnology of Sfax, which is the most relevant research center specialized in biotechnology in Tunisia. So the center develops cutting-edge research in all fields of biotechnology, industrial, food, environmental, plant, and health. Actually, CBS is organized in 6 laboratories and 5 units. So my laboratory, LPMC Research Laboratory, is the main laboratory at CBS working in the field of health biotechnology. And one of the strengths of the LPSMC is the complementarity of the skills that it brings together. So these skills cover key areas such as genomics, proteomics, and bioinformatics. LPSMC also includes researchers with skill in the medical and pharmaceutical fields to benefit from their expertise, covering the diagnostic of human pathologies, and to help to optimize the choice of and relevance of experimental therapy models. So since this year, our program is organized on 3 topics. So the first project, so project 1, the first project of the lab is interested to the identification and repositioning and valorization of biomolecule for therapy. So the laboratory actually currently has cell lines, microbial facility for cloning and ex vivo screening, but also as presented by our colleague, the zebrafish, as an excellent animal model. Donc, the laboratory have set up recently this model in the laboratory. The second project is related to genomics and the development of molecular diagnostic tools for the diagnosis of hereditary disease in Tunisia. And the most recent activity is digital valorization. The third project is the digital valorization of genomic data from control and patient and Tunisian patient. So thus far, our group of LPCMC, a grouc of LPCMC researchers, has largely contributed to the study of hearing loss. So we have contributed to several projects, national and international projects. But today I will present only our experience, local project developed in our laboratories. So hearing loss, which is the most common disorder, one of the most common disorders. So according to the World Health Organization 2019 report, over 5% of the world population have hearing impairment. Hearing impairment is usually classified or simplified as isolated, so or syndromic. Now, we have isolated deafness or hearing loss and syndromic, and there is a lot of syndrome related to hearing loss. And global genetic factors are responsible for over 50% of cases. As for several diseases, hereditary diseases, autosomal recessive inheritance is the most common genetic pattern for hearing impairment, in particular in countries with high rate of consanguinity, as in Tunisia and in North Africa. Hearing impairment is also characterized by high genetic heterogeneity. So today we have more than 121 genes only for isolated deafness. So the same phenotype, isolated deafness, only deafness or hearing loss, and we have for more than 100 genes. So which complicate genetic screening process. Among syndrome, I will just present Usher syndrome. So isolated deafness and Usher, among syndrome, I will present Usher syndrome. So Usher syndrome is inherited also as a recessive disease and is characterized by combined hearing impairment and retinitis pigmentosa. And there is 3 clinical groups, Usher 1, 2, and 3, that differ clinically related to the severity of hearing and retinitis pigmentosa, but also genetically. So today we have more than large 10 genes associated with Usher syndrome. So for a long time, molecular diagnosis tests for hearing impairment and in general for all genetic diseases in North Africa and in Tunisia have been carried out using routine screening techniques such as PCR-RFLP, for example, for this mutation, the most frequent mutation, 35-DLG mutation in GGB2 gene for hearing loss, or by Sanger sequencing for specific candidate genes. Later, in particular for hearing loss, and to address this complex genetic heterogeneity, we first designed a cost-effective North African deafness chip. That is a microarray that screens simultaneously around 58 mutations. So we used multiplex PCR coupled to the DL color array primer extension. So we include in this array all described mutation, the 58 mutation, and these mutations are distributed in 21 genes. At this time, in 2015, this is the 21 gene described in North Africa. So we include the mutation described in Tunisia, but also in North Africa, as we share the same founder and the same history. And usually the same mutation is found in Tunisia, in Morocco, in Algeria, and Libya. So with this technology, we improved the rate of diagnosis from 20%, around 20% to 30%. And later in 2015, we had really the opportunity to acquire at CBS the first next generation sequencing platform in Tunisia. is just a MySec. Just a MySec. Why? Professor Holden, in his presentation, present exome, the large exome screening as a routine screen. So the MySec, with MySec, we are not able to do the large exome. Why? With the MySec, the output is about 50 million paired and reads, so that yields about 15 gigabase of sequence, and thereby we are limited in the number of genes. So just several hundred of targeted region, not the large human exome, can be analyzed simultaneously with the MySec sequencer. But we have done, as I will present, a lot of work with this sequencer. So we have now the possibility with MySec to perform the large exome. So for our analysis, usually we define, we used custom NGS target enrichment library. And for that, and in order to investigate genetic in particular, as at the beginning of hearing loss, we used to design a custom NGS target enrichment library with the free short design of Agilent, is a web application of Agilent. And we assessed two DNA solution capture enrichment method, namely SureSelect and Alloplex, in order to compare technical outcomes, including depth, coverage, specific enrichment, performance in GC region, etc. This finding are guide usually laboratories in their choice of library enrichment approach. So the SureSelect was customized to screen 16 samples for more than 1,500 exon, with a total size of around 400 kilobase, and we selected around more than each time more than around 80 genes. The second, Alloplex. So for the Alloplex, we have the opportunity to screen in parallel 48 samples for the same size region, around 400 kilobase, and the number of gene is usually in our customized design is around 80 different genes. Just one difference between these two kites, that for genomic digestion, the first kites used a non-specific enzyme, topoisomerase, and the second use a specific restriction enzyme, and this impact the result, in particular the specificity of enriched region. So after SureSelect and Alloplex library sequencing, so this in this part, first secure the sequence generated by by the MySec sequencer, were usually analyzed with the SureCall. It's also a free web based for analysis offered by Agilent. Then we select variant according to allele quality, frequency, severity, inheritance, etc. So globally, with next generation sequencing, we improve more the rate of positive result. So from at the beginning 20%, 30% with microarray, and now more than 50% with next generation sequencing. Now I will present some results. As I said at the beginning, local results done in our laboratory in CBS, which illustrate the interest of the NGS, but also varied interpretation challenge. In his presentation, Professor Holden said that we used exome, it's more sample to interpret than using genome. I will, via my presentation, you will see we used some target gene to screen, so already confirmed gene as implicated in hearing loss disease, and we have sometimes difficult to interpret variants. Now the challenge probably is in relation with the interpretation of variant, and the interpretation of variant is associated with our responsibility or the responsibility of the medical doctor when deliver molecular diagnosis to the family. Now, so the situation I will present are could be met in any model of genetic diseases. You will see some cases are the same as presented by Professor Holden. So in the first example, using next generation sequencing, we identified in two consanguineous Tunisian family with a priori non-syndromic deafness. So the first exam, and here I come back to the remark of Professor Holden, to be associated with a clinical expert. So this family is addressed in our laboratory from our collaborator as having non-syndromic deafness. So we performed next generation sequencing. So two families, we performed next generation sequencing, and we identified one pair base deletion. Donc, to assess pathogenicity of variant, we usually use VARSOM, which is a freely community-driven project aiming at share global expertise between researchers over the world. VARSOM, the advantage of VARSOM that is offering database from at least 70 genomic database. So it includes data from at least 70 genomic database. In addition, VARSOM display automated variant classification according to the academic American College of Medical Genetics and Genomic guidelines. So every time each rule is explained, and we can modify if we have identified a novel mutation and we have an additional data, we can add it in the VARSOM, and we can modify the status of the variant. So here our variant is indicated as this delta mutation is indicated as pathogenic, and VARSOM define this variant as pathogenic according to here PVS1 because it's frame shift mutation, PM2 because here so it's the frequency related to this is moderate, so related to the frequency, and PP3 which is related to prediction by different program. So I come back to the ACMG guidelines. These ACMG guidelines published in 2015 are intended to be universally for all Mendelian disorders. However, criteria require gene or disease-specific knowledge. So it's different. To interpret variant related to hearing loss, we need to have a background with mutation and gene implicated in hearing loss. is the same for epilepsy and for each pathology. So we have CleanGen. Donc, CleanGen is an initiative from NIH. CleanGen established expert panel to evaluate gene and standardized variant interpretation by disease. Now, I am associated with the working group on hearing loss, and there is several working groups related to each disease. And according to CleanGen, is also we have the opportunity. So CleanGen is responsible of the to curated genes and variant. And according to CleanGen's WHRN, donc HRN is described as associated in particular to Usher syndrome. And the decision is definitive. That means that now you are sure that this gene is implicated in Usher syndrome. But when we look for non-syndromic, the conclusion for the moment is moderate. So this raises a question. As I said at the beginning, the family is addressed to us as have non-syndromic deafness. Now we have CleanGen, we have the expert group that classify mutation this gene as usually more probably associated to Usher and not to non-syndromic deafness. So we come back to the family. You know, in Tunisia, we do not all the exploration at the same time. So we come back to our collaborator, and we performed additional ophthalmologic exam, in particular electroretinogram, and we confirmed as showed here. So this is electroretinogram of our patient as compared with Usher patient. So we confirmed that there is no sign of retinic pigmentosa in our patient, and we confirmed that the family have no syndromic deafness. So this is very important to publish and to communicate to the CleanGen group, because when we accumulate results, this have an impact, and the decision of the hearing loss group can be modified from moderate to definitive. This is one of the family. As I said at the beginning, this mutation was identified in two families. Donc here, this is one of this family. Donc this family was published earlier by Emna. Emna will present just after me. 15 years ago, she published this family with mutation in the mitochondrial gene, one mitochondrial gene known to be associated with hearing loss, but also with mutation with the 30D gene mutation in GGB2. This is the most frequent mutation described in hearing loss at heterozygous state, but the mutation is at heterozygous state. But here at heterozygous state, sorry, but there is no mutation in these affected individuals. So we have identified mitochondrial in some patients. We have identified 35 gene mutation as heterozygous, and for some affected individual, we have no mutations. So 15 years later, and when performing NGS, we identify this mutation in the WHRN gene, and as it demonstrated here, all patients with profound deafness. So, because in this family we describe phenotypic heterogeneity. So, all patients with profound deafness have the novel mutation, excuse me, so this and this, and only this and this, okay, have the mutation at homozygous state. However, two patients with less severe deafness have only the mitochondrial mutation, the A155 in the mitochondrial genome. So this is just in the discussion, Professor Henry explained the fact that there is phenotypic heterogeneity because sometimes, in particular in consanguineous family, you have more than one gene. This is the case. This is the case. So in this family, we have at least 3 genes confirmed today as responsible of hearing loss. One mutation in the mitochondrial genome, which is associated, hearing loss usually associated with aminoglycoside treatment. We have the 35DLG mutation, but at heterozygous type, and we have a third mutation. So only with NGS we have the opportunity to have a real image about the situation in this family. And now our collaborator in the medical genetic department and the otolaryngology department could offer to this family a precise molecular diagnosis. Another example, and another novel mutation in CAP2 gene. This mutation, this gene is described in 2012 as responsible of non-syndromic and Usher syndrome. So you see that different genes are responsible in the same time of non-syndromic and Usher syndrome. So this gene was described in 2012 as responsible of non-syndromic and Usher syndrome, and the publication in Nature Genetics. However, when looking in details of the publication, and when we looked at additional paper published later, finally, the association between this gene and the Usher syndrome, the decision of the Clean Gene Group is refuted this association between Usher and the CAP gene, and only the association with the non-syndromic deafness is retained. So our family also have non-syndromic deafness, and this is also in favor of the decision of the Clean Gene Working Group of hearing loss. This is another example, a novel mutation also in PNPT1 gene. So we have also segregation. Using Varzom, we confirmed the pathogenicity of this variant. So pathogenicity is based on the frequency of the variant, on the prediction, and additional criteria. So is classified as likely pathogenic. And here we have to note that according to Clean Gene, this gene is associated with non-syndromic deafness, but also with Leigh syndrome. So the decision with non-syndromic is limited, and the association with Leigh syndrome is moderate. At the beginning, this family is also addressed to our lab as having non-syndromic deafness. So we come back to our collaborator to check, and finally we confirmed that there is no neurologic or other signs associated with leg syndrome in our family. So this is also a conclusion in favor of the implication of PNPT1 gene in non-syndromic hearing loss. So for the moment, we confirm what. Here, another mutation. This mutation was published earlier as pathogenic in an Algerian family. Now, this paper described the same mutation that we identified recently in a Tunisian family as a pathogenic. And their conclusion is based on prediction program and the frequency in Algerian population. However, when we looked at this variant in Varzom, finally, for Varzom, this the study association with this variant is uncertain significance. In particular, this decision is based on the fact that several missense variant described in auto-A gene are benign. So if the majority of variants described are benign, more probably for this prediction that also this variant is benign. There is other consideration for this gene to be considered, that this gene have a pseudogene. So when we have gene and pseudogene, so we need to be more carefully on the interpretation of variant, and this is the case. So for us, we have no co-segregation of this variant. So in our family, we identify the variant in the proband here, but finally and probably there is no segregation, as we have some affected individual at heterozygous state or presenting the wild type allele. So there is no segregation. Probably the difference here is related to another genes. So if we consider that there is no segregation, and as I said at the beginning, for Varzom we have the opportunity to add some data, some novel data. So here we added the fact that there is no segregation, and if we consider that there is no segregation, so you see that the state is changed from insignificant to likely benign. For hearing loss, I will finish with this example. When working on hearing loss, or other genetic diseases, we are usually in research of correlation between mutation and the severity of phenotype, and usually we have not this correlation clearly established. So in this example, we believe that probably we have some data in favor of the correlation of the mutation with the phenotype. So this here we identified a mutation in the kinase domain of myosin 3A, and when the family presented profound deafness, our family present profound phenotype, severe phenotype, and when we looked at the PubMed, we identified three additional mutations. in the kinase domain of mucin 3A. And we noted that for two mutation, the phenotype is severe. And for two other mutation, the phenotype is less severe. So we tried by molecular modeling to establish the correlation between the severity of the hearing loss, in particular in this case, and the position of mutation. And we conclude that if the mutation affect, in particular, the kinase activity and preventing the autophosphorylation of the motor domain. In fact, mucin 3A have a kinase domain and the motor domain. And the kinase activity is responsible of the regulation of the motor domain. So if you have a problem with this activity, there is no regulation of the motor domain. Here we found that we have a severe phenotype, as the mucin 7A will be moved to the tip, in particular, of microvelocity in our case, and will be accumulated in the tip of this microvelocity. In the other hand, if we have a mutation that destabilizes the protein, so we have or inactivate the molecule, we have a less severe. Why? Because we have the response, because there is redundancy of the activity between mucin 3A and mucin 3B. So different example here related to hearing loss. And now beyond hearing loss. Beyond hearing loss, we recently developed several NGS activities. First, as services for public and private laboratory, and we designed custom kits according to their needs for postnatal diagnosis of several genetic disease. But the most importantly now, that we also developed a novel research project in forensic DNA phenotyping in parallel to facial dysmorphology syndrome and intellectual disability. We also recently participated with Professor Tricky to a novel project, is a national but also international project on epilepsy. Also, we have developed in the laboratory microbiome activity, and more recently we performed for our ministry the sequencing of the SARS-CoV-2 virus. So for this novel project, I will present one example. This is the last example. So here we have two patients from a consanguineous family, two patients with hypotonia, nystagmus, psychomotor development impaired, dysmorphic facial, cataract, cognitive impairment. So association of several well, you heard me? Okay.
Yes. Yes.
Ah, okay, thank you. So in this family, we performed. So first we used the phase 2 gene. We used the first 2 gene, which is a deep learning based diagnosis system that utilize the aggregation of gestalt, so which summarizing feature of patient, to suggest candidate syndrome. So when using this phase 2 gene platform, several syndromes were suggested for these 2 patients. Among these syndrome, SLO syndrome, hyperphosphatasia mental retardation syndrome. So a lot of syndrome was suggested for patient number 1 and patient number 2. So as we have really no experience with gene implicated in this morphology, we preferred used site 1, which is a clinical exam. So around 5,000 gene can be explored in the same town. So the gene already described associated in OMIM with the disease. And using this site 1 sequencing panel, we identified a novel mutation in SRD5A3 gene associated with congenital glycosylation disorder. So if I come back to the suggestion of phase 2 gene in particular suggested at least for 2 patients, the SLO syndrome, which is a metabolic disorder, and hyperphosphatasia, which also is a congenital glycosylation syndrome. So here we identify our mutation in a gene associated with congenital disorder of glycosylation, but is another syndrome, and this syndrome for the moment is not included in the phase 2G panel. So we decide as the syndrome is not associated in phase 2 gene. So we decide to develop our customized recognition system based on deep learning. So we decide to develop a specific application, donc, to identify patient with dysmorphic feature, facial dysmorphic feature, based on 2D image. So first, in order to show that cases with this syndrome had recognizable facial pattern image, we performed a binary comparison between 13 healthy individuals and our patient, but also we included all photo of patient published with this syndrome. So as you see, we have identified 15 images from the different publication. And as we haven't a lot of image, we performed what we call data augmentation. So the data augmentation here, what that means that for each image was randomly augmented through rotation. So we rotate the image. So from one image, we will have several images. So rotation with a range from -10 to 10 degrees. We also perform a translation of image from in the x-axis and y-axis from -1 to 1. And finally, we used 70% of the image for the training, and then 30% of image for the validation of our system. So this is the result of the binary comparison that proved that patient with SRD condition could have a recognizable facial pattern distinctive from healthy control with accuracy around 50. So this is the excuse me. So around 97.5%. So in the second phase, and in order to evaluate the specificity of our syndrome, we included image from SLO patient and hyperphosphatasia mental retardation patient. So SLO and hyperphosphatasia syndrome as the common syndrome identified when we look at the beginning using phase 2 gene. This is the result, the first result. So this is the principal component analysis in 2 dimensions that displayed specific cluster distribution. So this is in green the SLO, the image of SLO patient. In brown, hyperphosphatasia patient, and here the SRD patient. So, as shown in this figure, we have different clusters identified using our system. Then, when we test for our 2 patients, so as shown in this figure, both are included in the cluster of SRD syndrome. This is another presentation of the result from our system. So here we evaluate by measure the accuracy of the top 3. So each time we test a patient and we see the result. So the result of our classifier system represents whether the diagnosis syndrome was first suggested in the shorted list. And as you see here, 22% for SRD syndrome. And for all the system, the accuracy is around 90%. So this accuracy provides potential in the use of computer assisted image analysis to identify a patient with the SRD syndrome. So currently we are also developing the application of this application on more than 10 syndrome. So we have a database of 2D image of more than 10 syndromes. So we have around 1,000 image, and we are trying to developing a system including syndrome already non-included in phase 2 gene. But even with the phase 2 gene, we have to note that phase 2 gene is prepared with image from Caucasian patient, and we noted that may impact the result when we use phase 2 gene. So developing a system using another architecture, but also using image from Tunisian and North African patient will be certainly useful. We are also now developing a novel project using 3D image, which probably can help to obtain more details from as compared with the 2D image. Yes, I finished. So thank you very much for your attention.
Thank you, Professor Sabar. Thank you for this wonderful presentation. from Tunisia, from Sfax. But we have some questions, but I prefer to have all the questions at the end of the seminar. And now it is my pleasure to present Dr. Mkawar, who she is an assistant professor in the Faculty of Science of Sfax and researcher in the molecular